introduction. So my talk today is about putting beneficials to work, which is a nice way of describing it, really, because that's what we're doing. Beneficial insects and mites and getting them to work in the vineyard for us. And as Melissa mentioned, we've been doing this for quite a while now. I've been here since 1999, which shockingly is 25 years. And this has been the focus of my research during that time. And the landscape in vineyards today is very different from what it was 25 years ago, when we depended a lot more on chemicals than we do now. We depend more on beneficial insects now than we ever have done before. 25 years ago, we hardly utilized them at all because we sprayed so much, they basically weren't there. So specifically today, I'm going to talk about two recent wine grape industry funded projects that are shown there. First one, improving biological control of grape mealybugs and spider mites in Washington vineyards using drones and an attractant. And the second one, well, the first one was a 2-year project, and the second one was just a 1-year project, which is an exploratory type of project looking at the prospects for using eugenol or clove oil as a possible attractant or IPM monitoring tool for the anagrous parasitoids of grape leafhoppers. And they're important biocontrol agents that we utilize to help in our management of leafhoppers. So those are the two projects. The first one, oops, gone too fast. The first one, using drones and an attractant to improve biological control of mealybugs and spider mites. We started that in 2022 with a partner from California, WAVEx, who are the drone people. I'm an entomologist. I know nothing about drones. I know a bit more now than I did then. But so they were our partners in the first year. And then in the second year, we had a local partner, GSL, GS Long, who you'll know, of course, who very kindly and generously provided their drone services. They're experts in using drones and have been for a while. So they contributed to no cost to the project, their drone services. And we also had BioBee as the company that supplied the beneficials that we were utilizing. So this project was a bit different in a lot of other projects I've done, in that we were trying to utilize purchased beneficials rather than the beneficials in the environment, although, as you'll see, we were using those as well. But BioBee is a company that produces beneficials, and there's a number of these companies that you can purchase beneficials from, which 25 years ago there may be just one company, I think, in Europe available for that sort of service. So that's a definite change as well. So getting some background research, that we've conducted at WSU Prosser on vineyard entomology over the last 2 decades has focused on conservation biological control, as I've mentioned already. That really has been what we've been all about, reducing chemical usage and getting the beneficials to provide more of their management abilities of managing pests than they did previously. So that's what we've been doing. And there's been a number of ways that we've done this, but two main ways that I'll highlight here are these. The first one is encouraging natural enemies of pests to become resident in vineyards by farmscaping, which is the term we use to provide the resources they need. So here on the slide, you can see flowers, native flowers in the foreground. And, all the beneficials that we utilize are, well, the vast majority are native. So they have existed here, for millennia, long before we were here. And they were associated with the native flora in this part of the world. So it makes sense that they still have strong affinities with native plants. So by utilizing native plants as being more part of our landscape around and in vineyards, then we can get these beneficials to be more resident and more effective, because we're providing them with refuge and also resources like nectar. You know, it's a well-kept secret, I guess, that most beneficials, even though they may be preying on insects and mites and, you know, doing biological control, they also invariably need nectar and pollen, to a greater or lesser extent, to maximize their potential as biocontrol agents. The second focus of our work over the last couple of decades has been looking at the use of attractants, and one in particular, which is highlighted here on this slide, not wintergreen mints, obviously, although we did try once putting out wintergreen mints. But wintergreen mints are basically methyl salicylate. That's the chemical that they contain. And methyl salicylate is a natural enemy attracting chemical produced by plants when attacked by pests. That's its main ecological function, or one of the main ecological function. Just about every plant species contains methyl salicylate in a greater or lesser quantity. And cut a long story short, we showed over a lot of years and experimentation that we could use this chemical as an attractant for improving biological control. I'll talk more about that as we go through this talk. Well, I'll talk a bit more now, actually. You know, we did a lot of trials between 2003 and 2011. We eventually got methyl salicylate in controlled release dispensers like this one here. They are now available commercially, called Predalure is the commercial name. And they're just basically plastic sachets that emit methyl salicylate daily during an extended period of time. And these are the kind of insects that it attracts. It's, you know, initially, you know, there's a lot of science behind this, and I won't go through it all, but initially we thought, you know, methyl salicylate as a chemical would attract certain insects. But to our surprise and delight, we attracted a whole community of beneficial insects, which seemed strange and hard to understand why this one chemical would attract such a diverse community as all the beneficials there. And this is just a selection of some of them, things like predatory bugs and predatory mites and lacewings and all the other things you see there. So we did some more research, and we found that this single compound methyl salicylate attracts so many because it's also a plant signal too. So when we put it out in the vineyard, we're not only attracting beneficial insects, we're actually telling the plants in the vicinity that pests are around. It's an early warning system. You know, plants talk to each other, and so once you know, a grapevine is attacked, it produces methyl salicylate, and its neighbor smells that, for want of a better word, detects it, and therefore knows that it needs to bolster its defenses because there are, you know, enemies around that will be attacking it. So basically, that's what it is. When we put in methyl salicylate in a vineyard, we're not only attracting beneficial insects, we're turning on the grapevines to produce their own predator attracting chemicals, volatiles, smells, if you like. And so that explains why we're attracting such a diverse community, because the plants are producing chemicals other than methyl salicylate. There's a whole range of other chemicals that are predator attractants, and that's what methyl salicylate will do: turn on the plants to produce turn on the plants to produce their own volatiles. So that's a little bit of the background to that. So that's, you know, all those two things, habitat restoration and attractants, were part of bolstering conservation biocontrol. But the current project, looking at drones, is a different type of biocontrol, augmentative biocontrol, which, as I've mentioned, you know, we're purchasing biocontrol agents from companies, and this is something that, you know, we can do and have been able to do for 20, 25 years or so. But, you know, and it can be a good tactic, but it has problems associated with it. First of all, you've got to buy them. You know, conservation biocontrol, one of the huge benefits of that is it's free, you know, largely free, because you're attracting what's in the environment already. So in augmentative biocontrol, you've got to buy them. But it can be a good tactic to support conservation biocontrol if you're getting the flare up for pests like mites, for example, flaring up. And conservation biocontrol, it is not a panacea. It doesn't, can't do everything. And if, there's an outbreak of spider mites happening because it's very hot in July, then conservation biocontrol may not be able to take care of it. So putting in, predatory mites that you have purchased might be a good way to control that flare up. But it is expensive. It can be expensive. Depends on the beneficial you're buying. And the other problem is, how do you distribute these things evenly? Since we're not talking about a natural process, we're buying a bucket of beneficials. How do you get them in the vineyard in an even manner? And this is where the drones come in, obviously. So augmentative biocontrol, adding extra predators early season. Predatory mites are one of the, the first natural enemies that you could buy from companies. And most of the success, the early success of augmentative biocontrol was from predatory mites, controlling spider mites. But now there's a lot of other beneficials too. But Gallindromus occidentalis, the western predatory mite, which is actually our native predatory mite in Vineyards here, the major one anyway. You can buy that commonly, and we use this native predatory mite in our drones, as you'll see, at least in the second year. But you can. The expectation is that augmented populations of predators will persist for at least a while, at least a season, we hope, giving prolonged enhanced biocontrol. But this is another one of the problems. And as you'll see in this project, we came across this problem again, is the fact that we don't really know if they are staying and maybe they're not. Things like this mite-eating lady beetle here has wings. And so, they disperse, they fly. And so they may not stay in the vineyard. Predatory mites probably more likely to stay because they can't, disperse as readily as a winged creature. But they can still move around in the wind. So this is one of the other problems is, are they going to persist for the time that we want them to? But if you're looking at controlling the flare up, you may not need them to persist for beyond a few weeks or so. So some other examples, lady beetles, parasitic wasps, predatory flies, lacewings. You can buy most of these now from different companies. So, there is a variety, and I'm sure some of you are actually using these already. And I know the Stepharis beetle, the mite-eating lady beetle we saw in the previous slide, is actually being used in hops early season to help control some of the early season hot spots of mites, and it does a pretty good job. And so again, using it for a defined purpose, using augmentative biocontrol for a defined purpose can be effective and cost effective. So this project, the mealybugs and spider mites, they both are good targets for augmentative biocontrol in vineyards because the mites are prone to flare up, as we know. Mealybugs, as we all know now, have a very low economic threshold. We don't want to see any mealybugs in a vineyard because of their role as transmitting agents for leaf roll virus. And so, conservation biocontrol usually, keeps, the whole point of that is to keep pests below an economic threshold. And most times an economic threshold is not elimination. And before leaf roll virus, we could have mealybugs in a vineyard up to a certain level without having problems. And so it wasn't, we weren't getting rid of every mealybug. But now we want to get rid of every mealybug. So, maybe augmentative biocontrol can fill the gap that the conservation biocontrol doesn't fill. And we also have resistant issues for both pests, chemical resistant issues. The chemicals we're using sometimes not as effective as we want them to. So they're probably good targets for looking at this strategy. And so the whole rationale was, could drones effectively and efficiently deliver biocontrol agents like these? and perhaps used for hotspot areas. So that's why we did the project. Flying up and down rows efficiently and quickly, dropping beneficials, potentially could replace a crew of 15, 20 people with all the associated costs. Cost of using the drone varies a lot depending on the species of beneficial insect and mite used. But you know, it's probably it's not going to cost more, and it has potential to cost less than doing it by hand. And probably more importantly, you're going to probably do, you know, we were hoping we could probably do a better job than having a crew of people out there delivering by hand. So this is a new project funded by the Washington Wine Commission back in 2022, and we did our first year with WAVex, the California company. And then in 2023, GS Long. And GS Long brought with them, I mean, you know, the technology of using drones, I've discovered, is evolving rapidly, and improvements are being made all the time. So our drones in the second year and the delivery was improved over the first year. The first year, you'll see in a couple of slides later on, the insects were just dropped out, not very gently. But this strategy technique here, the parabug delivery system that GS Long uses, provides a gentle action which protects the bugs during release, together with keeping the product and carrier mixed for more even coverage. But so there is a carrier like I can't remember what it is now, wood chips or sawdust, you know, which the mites and the beetles are mixed in, and so, you know, this is an improved way of delivering, and so I'm sure there'll be other improvements in the future, and probably the costs of using drone services will also come down in the future. So the protocols we had in both years, the beneficials released. Sorry, back again. The beneficials released. So for the mealybugs, we used the mealybug destroyer, which is a beetle up in the top right-hand corner. Cryptolemus is its scientific name, and it basically targets mealybugs. It might eat a few other things, but not much. It really wants to feed on mealybugs. So that sounded like a good thing. It's also, because it's a small beetle, pretty robust. So we think dropping it from a great height wouldn't cause it too many problems. So we used that in both years. In the first year, we used the predatory mite Neoceolus californicus. This was recommended to us by the California drone company because they'd used it before. This is the California predatory mite. And I think we've detected it here, but it's certainly not a common predatory mite in Washington state. But since we were, you know, not expecting it to survive the winter or anything, and it's, you know, summers here pretty much the same as in California, we thought it'd be okay. So we went along with that. But as you'll see from the results, that gave us less than good results. So in the second year, we went to our slightly more expensive predatory mite. So, you know, again, the expense depends on what you're choosing to release. The Western predatory mite, which is our native predatory mite that we most often find in Washington vineyards. The site we used is on Red Mountain, commercial vineyard with history of mealybug and spider mite problems. And the treatments are shown here. I can get this thing out of the way. So there was, in both years, in both years, but we had 2 drone releases of destroyers and predatory mites, which we timed to coincide with the abundance of early instar mealybugs after the eggs hatched. So that was, and we had strange years both years in timings. It wasn't what we were expecting. So in both years, I think it was about mid-June, and then the second release was in August, sometime early August, maybe mid-August last year. So that was one treatment. The drone releases. Second treatment was the methyl salicylate, which I spoke about earlier. So in the earlier work, we didn't resolve how few we needed per acre. We had some unpublished work that suggested, I mean, you know, at the beginning we were using hundreds per acre. We could go out there and it smelt like mints, you know. But we found that we needed a lot less to get better results, and we never really worked out how low we can go. But 25 per acre we felt did still work, so that's what we used here, and a treatment would last for 3 months. We also did the drone releases, the third treatment with methyl salicylate dispensers to see if we could improve upon either alone, and then of course the untreated control blocks. In the first year we had randomized 1 acre strips of vine separated by 50-meter buffers, and we learned after that first year that probably wasn't optimal because the buffering wasn't sufficient. A lot of the insects that we're talking about, the beneficials, are very dispersive, and we sort of saw that in our results, that sort of as time went on, there was interruption between blocks, did spill over into different blocks. We released 500 mealybug destroyers per acre plot and 50,000 predatory mites per acre plot. They were based on recommendations used elsewhere. Minimal pesticides were used in the block, and most of the treatments were safe to predators and done prior to the experiment. So in 2023, as I mentioned, we had the Parabug delivery system, which is that drone there. That's actually the first year drone, and you can see the hole that the insects just get dropped out of. But in the second year, we had better plot separation. We also used square plots, which I felt was much better too. On the other thing, we found that these beneficials we just purchased, they've traveled across the country, they've been kept cool, and then we throw them out of a drone, so into daytime heat. And so we thought that was less than optimal too, because a couple of the release days we were talking about 100-degree temperatures. So in the second year we had evening releases avoiding the daytime heat, allowing the beneficials to actually acclimate and find refugia in the vineyard, and hopefully survive better. And second year we used Gallindromus occidentalis as our predatory mite species, as I mentioned earlier, not the California species. And Predator, it's a methyl salicylate dispenser. These are available, as I said, commercially for $1.50 each, so they're pretty cheap. And if we're only using 25 per acre and we can get benefits from that, then yeah, that's cheap. So the rationale for using them, as I said before, is the fact that they can attract a diverse community of predators. A diverse community of predators is always very good. I mean, you can have specialists like the Cryptolemus beetle, but having a diverse community behind it is always to the benefit of stability of biocontrol. And we also figured that methyl salicylate could help retain the drone release predators. I mentioned earlier that one of the possible problems is that they could leave the blocks, the treatment areas, and so having methyl salicylate there might stop that a little bit. Data collection, these are the three major data collection strategies, which is mealybug trunk counts throughout blocks, sampling, random counts, leaf sampling, and sticky traps. These are three strategies that we invariably use when we're monitoring insect populations in orchards or vineyards, and they serve us well. So when the beneficials arrived, we wanted to check their viability, and that's one other thing I didn't mention, is the fact when you buy beneficials from an insectary, you sometimes really don't know what you're getting, because these insectaries are big commercial businesses, well, not big, but they're commercial businesses that rear thousands of beneficials, and sometimes it's not easy rearing thousands of beneficials, and you can sometimes get contamination or disease, or things can go wrong basically. So we wanted to check the viability, and in this first year with Cryptolemus we had so when they arrived, we kept them in the lab for 24 hours under optimal conditions, and just to see how they survived for the first 24 hours. So, Cryptolemus, we had nearly 100% survive. Neocellulis, the predatory mite, wasn't so good, down to 55 in some batches. But we're releasing a lot more of those, so probably okay. But in 2023, we had some shipments of beneficials that arrived all dead. So, this is one of the problems with augmentative releases. So if they, for some reason are exposed to hot conditions in transit, they might all die. And some other batches had very high mortality. So, this is always something to be aware of. And, work has been done in the past to show that some of the beneficials being supplied aren't actually the beneficials that they're supposed to be. So sometimes you don't know what you're getting, but hopefully quality control in these companies is actually improving. So some of these issues are being overcome now more than they were in the past. So that was assessing the viability before release. So we also assessed the viability after release. So this is, these are the Cryptolemus beetles being dropped from a drone at a great height. And then we collected some of them in trays within the plots. We went out, picked them up as soon as the drone dropped them, and then took them back to the lab and held them for 24 hours. And we found that they survived pretty well. So dropping them from a great height wasn't a problem, but which we didn't think it would be, but it's good to get confirmation. Okay, so now moving on to some of the data we got in that first year. The first thing that stood out from the data is that we had remarkable suppression of spider mites in all treatment blocks, as you can see in this slide here. The untreated is the bar on the right, and all the treatments, the far fewer spider mite per leaf than there was in the untreated. So that was good to see. The methyl salicylate, or the drone and methyl salicylate, and even the drone on its own, which was harder to understand, or not really, because the predatory mites, of course, we assumed were doing a good job. But the next slide shows that may not have been the case, because looking at numbers of predatory mites in the different treatments, you can see for the drone, there were hardly any there. And the few predatory mites we found, I believe, were not the Californicus. There were native predatory mites. So we concluded from that our Californicus releases were not effective. And really the reason for the spider mite depression was probably largely due to methyl salicylate. We know predatory mites respond to methyl salicylate. They're able to orient towards methyl salicylate. And more importantly, methyl salicylate is attracting a lot of other spider mite predators too, like the Stepharis beetles, which were there in great numbers. So we think most of the spider mite control in 2022 was mediated by other predators. But it certainly looked impressive in terms of spider mite control. Mealybugs, reduction, we did get reduction, but nowhere near as much as the spider mite reduction. 13 to 20% reduction was seen in the 2 drone treatments, slightly better with the methyl salicylate. Yeah, so that was disappointing. We had a reduction, but it was nowhere near as great as we were hoping. So we had, that first year gave us rather mixed results. So in 2023, we were hoping with the new design, it will not new design, but new location with better plot separation, which might have been part of the reason for the lack of results in the previous year. We were hoping for better things. So with spider mites, we still had what we had in the previous year, which was good to see, a greater than 50% reduction in spider mite populations in the drone and the drone plus methyl salicylate treatments. So that, we had that in both years. And then the predatory mite population, again, unfortunately, did not appear to boost predatory mite populations above what we saw in the control. And so again, our releases of predatory mites via the drone may not have been that effective. We had most predatory mites again in the methyl salicylate treatment. So that was slightly disappointing. That maybe our predatory mite releases were not doing very well. And one of the problems of this is knowing, as I alluded to earlier, knowing the survival of these beneficials. You know, we did our tests a day after release and those sort of things, but we want to know how they survived, many days after release, or a week after release, or a couple of weeks, and it's hard to get an indication of that. I mean, there are ways of doing that, and maybe we need to look at that in the future. We could put dye on them or something that would identify them from other predatory mites, and also and look at them more regularly over time. But at this point, it does seem that the predatory mites, at least, were not doing that good after we released them. So the mealybugs, looking at trunk infestation, we had again a reduction, but not as great as we wanted. 33% approximately in the drone only treatment. We didn't get much of a reduction at all in the drone and methyl salicylate treatment, nor in the methyl salicylate treatment. So again, that was, we got a reduction, but it was not anything to write home about, unfortunately. Next slide. Looking at mealybugs in another way, and this was looking at the sticky traps actually helped look at mealybug numbers by trapping female and nymphal mealybugs as they walked around the vines, which is, we weren't anticipating that, but we got large numbers from that method, and using those data, there was a 37% reduction in the drone treatment, and all the treatments were less than the control. So we are getting some reduction, but not as great as I was hoping for. And looking at sticky traps again, for mealybug destroyers, we found most of the mealybug destroyers in the drone treatment, as we would hope. But so superficially this graph looks impressive, but the numbers are very small. We only had 5 mealybug destroyers during all of our sampling in the drone treatments. So clearly, using sticky traps to sample mealybug destroyers after release is not a good way, and some insects are not good to sample, or they're not effectively sampled by sticky traps because, for whatever reason, they're not attracted to the yellow trap, or they're not stuck to the glue enough. We don't know. And also looking at the numbers of mealybug destroyers on leaves gave us similar low numbers. So and they drop off of leaves when you pick them. So that's clearly not a good way. We could go out and beat the foliage. We used to do that years ago when we were monitoring some of these beneficials that are very mobile, and that might give us a better idea if we just went out and shook the canopy and collected what dropped off. We might get a better idea of the survival of Cryptolemus that way. So like with the predatory mites, we don't really have a good idea of how well they persisted in the treatment blocks afterwards. Our sticky traps also showed us good suppression of leafhoppers, not a pest that we were targeting by any means, but again, this might be due to the benefits of methyl salicylate primarily, and maybe even the Cryptolemus feeding on immature stages of leafhoppers. That's not impossible either, but it was interesting to see that leafhoppers were also suppressed. And then looking at some of the beneficials in these blocks after our treatments, some of the other beneficials, the conservation biocontrol already present in the habitat beneficials, you can see spider mite destroyers, the other type of destroyer. There were huge numbers in our treatment blocks, including the drone block, which is a bit hard to understand, but mostly in the methyl salicylate and the drone plus methyl salicylate blocks, where we had more than double the numbers of these very effective spider mite predators present than in the control block. So clearly we're doing something right in terms of attracting beneficials and getting them to go to work by the treatments that we applied. And the same thing with we look at all the beneficial insects by treatment. There was far more in the treatment blocks than in the control block. So from that point of view, it was satisfying to see that we were able to bring more, beneficials in and to get a better response from them to a large extent. So briefly, the outcomes. Well, we've shown that drones can be used successfully to release predatory mites and beetles. It is an effective way of doing it without harming them. And the way that the drones can navigate blocks makes them highly usable in this context. And so if there was just a targeted area, an outbreak that you needed to target, they'd be ideal for doing that, or to do the whole block more effectively and efficiently than using a gang of people. So the releases of mealybug destroyers did suppress mealybugs by between 13 and 37% over the 2 years, as far as we can ascertain with the data we've analysed so far. We've still got a bit more data to analyse. But the degree of establishment by the beneficials we're releasing is still uncertain. And if we do more work on this, we need to look at that. A methyl salicylate suppresses spider mites and leafhoppers by more than 50% by attracting predators, spider mite destroyers, lacewings, lady beetles. We've got a lot of data on all these other predators too. And we're seeing a lot of attraction, just as we saw decades ago with the use of this very effective compound, methyl salicylate. So, methyl salicylate does boost beneficial insect and mite populations. And I hope to revisit the whole idea of using methyl salicylate in the future. I have a proposal in with the industry now to see if we can do that, to see how low we can go in terms of putting out dispensers. I mean, it's already fairly cheap, but we might be able to go even lower. And also what happens if we use them over multiple years, because in the past we've only ever used them as one-offs, and so it's likely that the benefits are going to improve and sustainability will improve if they're used on a routine basis. So that's exciting to bring that back again, because I truly believe that methyl salicylate is a very valuable component of can be of grape IPM, and the fact that it is available commercially, I think just makes it even more logical that we should be using it. So the drone releases of beneficials can improve biological control, and the degree of improvement likely varies according to the type of beneficial. We could get better results if we use lacewings, and lacewings have been used for mealybug control elsewhere in other crops, and so that would be worth looking at. The cost effectiveness, we'd need to look at that as well, but the prospects are pretty good for them being cost effective. And the methyl salicylate dispensers, as I've already mentioned, show great potential, and I hope we can look at that more in the future. Okay, so moving on to the second part of this. No idea what time I'm taking over this, but this will be shorter because this was a 1-year project. Looking at the potential of using eugenol or clove oil as an attractant or monitoring tool for the parasitoids of grape leafhoppers. And this picture here is one of those anagrous parasitoids. And they are egg parasitoids, so they're tiny. A leafhopper egg is tiny, and the immature development of these wasps takes place within one egg. So you can see how small they are, but they're tiny but mighty. With the numbers that they can produce, they can produce very good biological control if they are present in vineyards in the first generation of leafhoppers. So the quicker that they can get into vineyards, the better the biocontrol is that they provide, because they can develop quicker than leafhopper generation. So they can have multiple generations to one or two leafhopper generations. So a lot of our work in the past was focused on getting anagris in early. This slide I found online, which illustrates how small they are. That's a coin, probably a cent or something. And so you can see how small these anagris wasps are. They're barely visible to the naked eye, but they are very valuable. And we don't see them, but they're there, or they should be there. So anagris wasps, and those are eggs where the anagris is developing within them, and a high percentage of leafhopper egg parasitism in spring can lead to good summer control of summer leafhopper population. So, not getting rid of them, but suppressing them below outbreak levels, below levels that, cause problems with pickers getting them in their eyes and such like. So we have a complex of anagris species, and they're listed there. So at least 4 species, there's probably more, but they're the 4 most important. And the first one, Atomus, is probably the most important in terms of the work that we did in this project. Yeah, so a lot of the work in the past was based on trying to improve anagris overwintering. So by growing roses nearby to vineyards, you're providing an overwintering habitat for anagris, which means that they can get into the vineyard early. So we did a lot of work at that time on anagris because it was important. It felt, it was a good way of improving biological control of grape leafhoppers. But since that time, we haven't really gone back to look. I'll go back one slide. Gone back to look at it. Why am I not going back? So in 2021, I came across this paper, which is looking at the, go back, looking at a sex pheromone in Anagris atomus, which, as you remember, was one of the species that I had listed there that occurs in Washington. And they identified the sex pheromone as eugenol, another common chemical that's readily available. And this species apparently uses, the females use it to attract males, and they demonstrated this in the lab and in the field. And so because it's one of our species, I was interested and excited by this, because maybe we could use eugenol to attract this species, and if it's attractive to this species, maybe it's attractive to other anagris species, because that's often the case. You have a genus of insects where the pheromones are overlapping, and sometimes there won't be total overlap, but there's usually some overlap. So it certainly felt like it was worth investigating, just as a quick project, to see if there was any potential, to see if we could monitor these tiny little biocontrol agents in the field, because it's hard to monitor them because of their size. You can only see once they're there and doing their job, whether they're present or not. It would be good for us to know a bit earlier than that whether they were around. And so if we had traps out with eugenol or clove oil and could trap them, that might give us a good way of monitoring the population to see whether you had anagris in your vineyard. So this chemical is eugenol, readily available, fairly cheap. It's also the major ingredient of clove oil, which is even more readily available from supermarkets, I imagine. So that was our objective, just a simple project to look at the attraction, if there was any, of these compounds to anagris parasitoids of grape leafhoppers. So we planned to start it in August, September 2022. Actually, we planned to start it earlier in the spring, but we couldn't find any anagris. And that was the first warning sign that we had that something different was going on with anagris, because it's been a decade at least since we monitored anagris populations. So we had difficulty finding anagris populations in vineyards. So by August, September, we were getting desperate. And so we started looking around blackberries, which were our old haunt for looking for leafhoppers and anagris, because there seems to be some movement between blackberries and vineyards between, by leafhoppers and their parasitoids seasonally. We discovered that years ago. So we went back to the blackberries, and we did find numbers of anagris there, but they were small. Best numbers 10 to 50 per trap per week when we were expecting hundreds. But at least we found some in blackberries around Prosser. So we tested a range of concentrations of these two compounds diluted with hexane. We put the solutions in small vials and attached them to sticky traps, plus a control treatment, which is hexane only, and ran the traps weekly for a period of time while the anagris was still present, which up until mid-October. And then we brought the traps into the lab and examined them for the anagris wasps. And so here are the results from that first experiment over about a month or so. And you can see with eugenol, there was no evidence of attraction. The control bar on the far right-hand side always seemed to have more anagris than the other treatments. Maybe the best was the lowest treatment. I mean, we had no idea really what kind of concentration rate we should use. So we just thought we'd start at the top and work our way down. So with eugenol, we saw that 0.1% possibly offered the best potential. With clove oil, same range of concentrations, and again 1% and 0.1% appeared to be better, and in fact better than the control in that particular thing. So that was moderately interesting and potentially exciting. So we had to give it up for that year and then go out the next year and extend the project into God, I can't get that again. Extend the project again into the next year. And again, we had trouble finding anagris populations early season. And that turned out to be important in the end, because the Atomus species, which was the species that, is documented by the European research as showing the pheromone attraction, only occurs, looking back at our data, mostly occurs in our vineyards early season. Later on in the season, other species join the party. So we really would have liked to have done this experiment early season, since we knew that would be the species that the Europeans had shown was attractive to these compounds. But we had to do it later in the season, because that's the only time we found substantial populations. And this was at one site, an organic vineyard, where we had numbers in the thousands, actually more than 1,000, well, up to 1,000 per trap per week. But, over the whole vineyard, there were thousands. And so this was reminiscent of what we saw a decade ago. So we were able to do our trial, our little experiment. We tested a range of concentrations, as you can see, from a lot of zeros, 1% to 0.1%. So going down from the previous year, same methodology, except this time, in the previous year, we could just do single traps because we had very little population or area to work with. But this time we had a bigger area, so we could replicate things and make things statistically better. So yeah, we conducted the trial during that period of time, August to October last year. And unfortunately, the news is not good. We had high numbers of anagris, very robust populations, as you can see from the side there, up to 1,200 individuals. So a lot of anagris to count on the traps, but we didn't see any more in the treatments than in the control generally. Sometimes the treatments got close to the control, but there was never any more. It seemed that the higher concentrations, 10%, 1%, whatever, we might actually be repelling anagris. But so and so it was the same with eugenol. I think the other slide was clove oil, and so with eugenol it was the same. And we went down even lower with eugenol because there was a flicker of hope when we started getting lower concentrations, but that didn't eventuate. Control always beat out the treatment. So this may have been because of the different populations of Anagrass species we had, which were the three other species, not Atomus, and the possibility that the pheromone is not the same in those species, or there could be other reasons. But the most likely reason that it's a sex-specific pheromone for Anagrass atomus, and our other species dominate during the fall. And the only way to check that would be to find spring populations that we could test. And so and if we want to pursue this further, we'd need to identify the sex pheromones of our other Anagrass species. We could do this in the lab if we could rear Anagrass, and we probably could rear Anagrass. It would be a more complicated thing to do, but it's certainly doable, as the Europeans did. And if we were really interested in pursuing this, that's probably the best way to do it, to see what our other Anagrass species are responding to. But probably the bigger concern to me is, sorry, just this last slide, Melissa, is I can't move this block thing. I'm having trouble with my slides here. Sorry about that. It highlighted a potential problem with the functioning of Anagrass in our current vineyard ecosystem, because Anagrass seems to be uncommon in vineyards, but more uncommon than it was in the past. We did a lot of searching around in vineyards that we would expect to find Anagrass and didn't find it. So there's a number of possible reasons why. There might be a possible shift in leafhopper species abundance. We used to have Western grape leafhopper as the #1 species and Virginia as the #2. That may have shifted, and we don't have any data on that, but anecdotally that's a possibility, and that might affect Anagrass, because Anagrass doesn't seem to like Virginia creeper as much. So that might affect parasitism. And also the use of pesticides now, particularly neonics, that we use a lot in vineyards, have a known suppressive effect on a lot of beneficial insects over time. And so we may just have been removing Anagrass from our vineyards, because it is interesting how we only found the large population in an organic vineyard. So this project, if nothing else, has sparked an interest in knowing what is actually going on with Anagrass and biocontrol of leafhoppers. And so again, I hope, like with the methyl salicylate work, to pursue that further in the future, to find out what is actually going on. Thank you for listening to me, and if there are any questions, I'll answer them.
Yes, David, we do have one.
The people I thank. Thank you.
Yeah. And I do want to give a big shout out to Aaron Avila with GS Long. He was really key in coordinating everything with David. And so thank you so much to GS Long and BioB for giving us a break on the beneficials. So there's one question in the Q&A. How long after release were the predatory mites sampled? And then did you look for pest mites sampling at the same time when you were in there counting your predatory mites?
Yes, I think the first sampling date was 2 weeks after release. And at that time, at the release time, spider mites were not really present in any great numbers. They occurred later, at least from the first release of the beneficials. So ideally, probably we should go in at least a week, after release, and maybe even just a couple of days after release. That would be instructive in terms of the beneficials we're releasing. Even a day, and we could do that. We could, do for the first few days, do sampling, and in retrospect, we should have done that.
And then another question for the predator sachets. How many man hours were required to put them out there per acre?
Well, well, you're putting out 25 per acre. Growers probably know better than I do how long that would take, but you know that's something else we need to look at, whether they need to be evenly distributed. I think they probably would, but that's not necessarily true. And there's potential that we could get it down to 10 or even 5. So you know, I mean, it's certainly not as labor intensive as distributing beneficials where you've got to go to every vine. So I think it's not going to be a huge issue, and you're only doing it once or twice a season. So there you go.
Okay, another one. Did you see any Cryptolemus larvae in the plots?
No, we didn't. We didn't see any larvae at all, which we should have done on the on the leaf samples or on the vines on our trunk examinations. Oh, sorry, we did see a few, but nothing to write home out on the trunk samples. But you know, I was expecting to see them on the leaves as well, as we have done in the past. But so again, that doesn't bode that well for their continued survival after we release them.
Yeah, and then again, the rate for the methyl salicylate dispensers per acre that you used in 2023, that was the same rate of 25 per acre?
Yes, correct.
Okay, let me see if I have any more. Oh, a couple more. Does the methyl salicylate attract anagris?
Yes, indirectly. Again, it's working on the principle I explained earlier, where the methyl salicylate is causing the vines to produce other chemicals to attract things, and one of those chemicals apparently is anagris. So yes, we have seen increased anagris populations in the presence of methyl salicylate dispensers. We don't have it well documented, but that is another one that we're pretty confident it does do, and we need to document that further in the future.
And then if you were just using the sachets, the Pred-Alert, what time of year would you want to get them out there? How
Again,
early?
that's something we need to look at. In this experiment, these trials, we didn't put them out till May, which I'm beginning to think is maybe a little late. I think it's more optimal to put them out in April and have them go 3 months, and then put another set out at the end of that 3 months. They last for 3 months with their release rate. And I think a season-long exposure would be better, because if we, even I keep forgetting we're turning the plants on. And so, the sooner the plants are turned on, the better. So we can turn the plants on once there's leaves there. So yeah, I think in any future work, we'll be putting them out in beginning of April.
Okay, any other questions? That's all I see. If not, thank you all of the attendees for giving up your lunch hour and cutting into your early afternoon, especially on such a great day. So we will end and let you get back out to the vineyards and get back to work. So thank you very much, David. And David will be finalizing his reports at the end of June. And then once we have those written reports, you'll be able to find them in the Wine Commission's research library. There'll be 2 separate ones because they were 2 separate projects. So that's always a good way to refresh your memory. And then also we will be putting this webinar recording up on in the research library as well. So, and you all will get a copy of it just within a couple days. Make sure I didn't miss anything. Oh, yep, and that's it. Thank you very much.
Thank you, everybody.
Okay, bye.
Bye.